But today's lesson might be a good one to miss, even though I don't want you to walk out. This one's a pretty easy lesson because it's basically some functional anatomy of the kidney that I want you to know. And I hope a lot of this is review from some of your past semester gross anatomy lessons or some of the histology lessons that Dr. Meek gave you. But I would expect you to know basically how to describe the kidney right now from an anatomical point of view. I think when I mentioned the words like kidney cortex, you know there is a cortex of the kidney, a medulla of the kidney. In the human kidney we have an outer medulla and an inner medulla. The functional unit of the kidney I think you know is the nephron, and I think you went over a number of cell types of the nephron with Dr. Meek, with the proximal tubule. Okay, our human kidneys and mammalian kidneys have this loop of Henle that starts in the cortex of the kidney up here, and the medulla is down here, so that our nephrons actually have this loop, dips down into the medulla, and then loops back upward to the kidney. That's called the loop of Henle. I think Dr. Meek went over that in some of the morphology of the cell types in there. So the proximal tubule, you know, if you had to describe the nephron, I think you could describe some of that. Am I correct? Okay, you know about that loop, you know, the thick ascending limb, that rings a bell. There's some cell types in there that are very important. A distal convoluted tubule that has some specific cell types, and then a collecting duct. And actually the collecting duct comes from a different embryological origin than the other parts of the nephron. But then the collecting duct then starts in the cortex of the kidney and goes all the way down to the bottom of the medulla, begins to coalesce with some calyces, and our urine begins to collect down there, goes into the major calyx, what's called, and then finally the initial part of the ureter, and it goes on down to the bladder. So the basic structure of the nephron we have to go over, and some of the morphology of the cell types in the nephron. I expect you to know the basic morphology of the proximal tubular cells, thick ascending limb cells, thin descending and thin ascending limb cells. We'll go over that. Thick ascending limb cells are very important. Function there is very important. Distal convoluted tubule, and then our collecting duct. I think you've heard of the principal cells and the intercalated cells before, right? I think Dr. Meek kind of went over those. So there's some basic transport characteristics of those cells. When we talk about what the kidney does and how the kidney does it, we have to talk about these specific cell types and their function and their transporters. So the nephron in the kidney is very heterogeneous, okay? Different cell types with different functions, different morphology that serves those functions. Now, mainly for the most part, we'll talk as if the kidney has one big nephron, but that's not the way it is. Our kidneys have about 1 million nephrons, and they're different sizes, different shapes, all the same basic structures, but they are different. And basically we divide these into two separate types, one called a superficial nephron, and the other is a juxtamedullary nephron. Three basic differences I want you to know about those with regard to the glomerulus size, the loop of Henle size, and the location of the glomerulus. Okay, superficial nephrons have their glomerulus close to the surface of the kidney. They have short loops of Henle, and their glomeruli, or their renal corpuscle, is small. Juxtamedullary nephrons have very long loops of Henle, large glomeruli, and they're close to the medulla. The filtration that occurs in the kidney occurs at the renal corpuscle. So when we go from the blood into the kidney, this process of filtration is something that I'm going to cover in detail tomorrow and the next day. And it's one of the basic fundamental functions of the kidney. And this is where a lot of diseases occur. So the filtration of the kidney, if you had to describe that based on what you know now, what would you say? Well, our kidneys filter from our plasma into the renal corpuscle, or Bowman's space. I have to stay over here because the microphone's over here. And there's a filtration barrier that I want you to know the structure of. And basically there's three things there I want you to know: the endothelial cells, the basement membrane, and the podocytes. I think Dr. Meek probably talked about podocytes, and I think you probably saw more EMs and histology slides of renal corpuscles and glomeruli than you could probably stand. But that is a very important part of renal physiology, so the filtration barrier is something I expect you to know about. And finally, this JGA, or juxtaglomerular apparatus, which is a very important part of the kidney, is it includes granular cells that secrete renin. These are part of the afferent arteriole. It includes extraglomerular mesangial cells and the so-called macula densa. Okay? And that, I think, should ring a bell in your head as well. Okay? So these basic things in functional anatomy of the kidney you need to know now, so that when we talk about them in more detail, you'll know what I'm talking about. So the gross anatomy of the kidney, you need to know basically cortex and medulla. The renal vasculature, what is unique about the microcirculation of the kidney compared to the microcirculation that you learned in cardiovascular physiology from Dr. Benjamin? Okay, if you remember, in your microcirculation, you've got your arteriole, and then it branches off into this capillary bed where you have filtration outward and inward, and then it coalesces into what? The venule, okay? And then it goes on into the venous system. The kidney is different. The microcirculation of the kidney is unique. We have an arteriole called the afferent arteriole, goes into these glomerular capillaries where filtration occurs. Then it coalesces into another arteriole called an efferent arteriole, and that then coalesces into another set of capillaries. So the microcirculation of the kidney is extremely important, and we'll go over that. The nephron segments I just mentioned, superficial versus juxtamedullary nephrons, be able to distinguish those. You might see an exam question about that. And the renal corpuscle. Okay, this is where filtration occurs, where you have your glomerular capillaries and the beginning part of the nephron, which is called the Bowman space. So all of this plasma, water, electrolytes, non-electrolytes go from the plasma into Bowman space, and this is where the kidneys begin to do all of the things that they have to do in order to process this fluid. And then we have the juxtaglomerular apparatus.